as well. Okay, great. It's live now. Thanks, John, for confirming. Yeah, so good morning, everyone. Welcome to the third lecture for Projects and Seminar Mobile Genomics course. So we explained in the previous lecture these three main points: what is genome analysis and why we need it to be intelligent, especially in the pandemic time. And we briefly describe how we analyze genome. For today, we will go through different topics still related to the previous lecture, and that is including read mapping. So we know that as a result of the shortcoming of the sequencing machine, we are not getting the full genome sequence, but rather we got subsequences of our DNA, and that's what we call read. So we need to link these three together. That is what we call read mapping. And so read mapping is a result of the problem that we have in the sequencing machine. However, this read mapping is still a bottleneck in this pipeline, and we will explain the reason why it's slow. And then toward the end, we'll start introducing the algorithms and hardware acceleration, especially from our research group at ETH Zürich. And I think we will not have time to go through details, but we will keep this topic for the next section, for the next lecture. If you have any question about the previous lecture, just let me know. Otherwise, yeah, by the way, the slides are also available in the course page right now. You can access them, especially for those watching us on YouTube. So if no question, we can start directly with what is read mapping. So I hope you still remember this from the previous lecture. We got the DNA sample, that is a blood or whatever, and we do processing in the lab to isolate the DNA from all other molecules in the cell or in the sample. And then we send it to the sequencing machine. We got the reads. These reads are, again, have no any information about their location or their order. And for that, we need a reference genome. We try to map one read or one piece of these pieces to the reference genome, finding the optimal location. And then we do the same thing for all other pieces until we hopefully get back our complete DNA. That's what we call read mapping. So read mapping is to map reads to a known reference genome with some minor differences allowed. Now, why we allow these minor differences? There are two main reasons. The first one, the machine are not perfect as any other machine, so they introduce sequencing errors. These errors, like there is no confidence about reading one base in your DNA. So the machine can say this base is A, but in fact it's C, something like that. So we need to tolerate these in order to enable correct read mapping. Also, the second reason, which is the goal of all this entire analysis, is to find genetic variation. And these genetic variation can be insertion, deletion, substitution, or many more large genetic variation types. And for those, we need also to tolerate them such that we are able to find them at the very last step of genome analysis pipeline. Okay, so again, I'm going to explain this analogy, which I like it very much, and we already explained it in the previous lecture. So the reference genome, you can think about it as the picture in the puzzle, and then the reads are the pieces that you want to solve the puzzle for. So you have these pieces, you try to compare them to the reference picture, and hopefully you can find a correct place for that, and then you can build your puzzle. Okay, so now where to get these data? Where to get the reference genome? Where to get these pieces, or what we call reads? So first, the reference genome, we are lucky enough to crack the first human genome sequence in 2003. That is an effort of 13 years, and it cost a lot. So every single character in the reference genome cost $1 to read it. So you can think about the human genome is about 3 billion character. That is $3 billion. It's really a lot in that time. And this effort is really a collaboration between different countries, including UK, US, Japan, and many more. So it's really expensive to build or to find the reference genome in that time. So that's why we don't have enough number of reference genome in that time. Okay, so however, the reference genome we got in 2003 was not complete. So we only able to get a nearly complete human genome in this year, in May 2021. So it was really an effort of 3 decades, not only 10 years, and yet to be complete. So this is a new paper. Again, this is consortium. So it's a large effort from many research group, not single research group, try to working hard to complete the reference genome. And they could find 200 million new character in our reference genome to close some of the gaps that they were complex to be sequenced in that time, in 2003. However, still we have some gaps, especially in the Y chromosome. For example, we don't have the full sequence until now for the Y chromosome. But anyway, we can still use this reference genome, which we're doing good, especially for many other applications that were enabled by this nearly complete reference genome. So if you are interested, you can check this paper. You can see the link over there, what enables finding these gaps or closing these gaps. Because of the advancement in the sequencing technology, we finally are able to do something to the reference genome after 2 decades of research. Okay, so you can download the reference genome from this link. It's publicly available on NCBI server. You can download the file. You will get the file something similar to this. These are the first first few lines from the file. You can see it's NNNN. It means the sequencing machine cannot read these bases. But after that, you will start seeing a lot of ACGT's characters. Okay, how long this DNA? So for viruses, normally it's very short, and normally a few thousand characters. But for bacteria, it's a few million characters. And for a human, as I said, it's around 3.2 billion characters. However, we are not superior to other species. So for example, red onion has much more DNA than we have. So they have longer with 16 billion, that is about 5x longer DNA than ours. And this Japanese flower has about, I think, 50x more characters than our DNA. So you can see that the workloads really varies depending on the species, but plants and fruits are well known for having very long DNA. Okay, now we finished from the reference genome, assuming we got the reference genome for the target species. And now where to get the reads? So either you can do the sequencing yourself, as I explained in the last lecture. You can buy one of the commercialized kit from a pharmacy or supermarket, and then get the sample yourself, send it to the company. They will do the sequencing for you. Or you can get one of these publicly available data set, for example, again, from NCBI data set, like SRA or ENA in UK. So you can get one of these read set. You can see here, this is the link. If you access it, you will see this is one of the data set that is produced by this machine, Illumina HiSeq 2000, that is producing short reads. So the reads are limited to around 100 long characters. And you can see more information about the sample, the characteristics of the read, and so on. Okay, now we assume we got the read set and we got the reference genome. How we do read mapping, how we find the location of each read in the reference genome. Let's learn how to map it. So the brute force algorithm is just to try every single location in the reference genome. Hopefully you can find a good hit while you are visiting this very long path. So we pick the read, we have the reference genome, we try to visit every single location and check, does it match, does it match, does it match, until we find a good hit. That is a brute force. However, it's very expensive. And this is the time complexity to do that. Imagine if the reference genome, which is the N here, imagine if it is really huge, like 150 billion character, as in the Japanese flower, for example, then you will spend a few months to do the read mapping. For that, we really need to do very efficient read mapping. And I would like to encourage everyone to check our recent paper, just published the last month in Genome Biology, about how the sequencing technology dictates algorithm. How, based on this sequencing machine, we are developing new algorithm whenever we have a new machine. And this algorithm trying to be very efficient such that they can cope with the workload coming from the sequencing machine so that we enable scalability of the analysis. So again, as I said, the time needed to do single analysis is not as important as how many analyses I can do in one day. I don't want to wait sequential time to do 100 samples, for example, but rather I want to enable parallelism or to enable much more scalable such that I can do analysis for the entire population or the entire city of Zurich, for example, in one day. So as I said, this is a very recent paper, and this is what we got from the bioinformatics community about the paper. So hopefully this will encourage you to read the paper. We got some tweets in Espanol, but this is very encouraging. I would like to thank everyone in the community for their feedback as well. Okay, so this is another analogy to do read mapping. I come up with this analogy and I like it very much because I was using these yellow pages when I was a kid. So I think it's very similar to looking for a phone number in these yellow pages. For those who doesn't know the yellow pages, this is a book where all phone numbers and citizen of a city are recorded in this book. So if, in the past, if you want to figure out a phone number, there is no way to record it in your smartphone because smartphone did not exist in that time. I'm talking about 90s. I'm not that old, but yeah. And in the past, we were using these books called Yellow Pages. Maybe they have other names in your country. And to look up for a name, you really need to go through the index, the very first pages, and these names are sorted in the index. So you go through the first character until you find the target first character, and then you go to the next character, and then the third character until you find the full name, and you can search it in the pages and get or fetch the phone number from there. And that's exactly how we do read mapping in reality. So first, get the page number from the book index using a small portion of the name. So normally we use the first character or the first two characters to get the page number from the book. And then we go to that page, retrieve it, and then try to find the full name from there until you get the phone number. I hope this analogy is clear to all of you, so that I'm going to explain now step by step how we do read mapping as algorithm. Okay, I assume it's clear. Let me see if I can see the faces. Yes, now I can see it. Okay, so now let's go through the algorithm. We have the FASTA file. We have the FASTQ file. The FASTA file will look something like this. Remember, this is the reference genome. So the first line is just the ID of the reference genome. The next few lines are the content of the reference genome. So all of these are single line, all of them here. Now for the FASTQ file is different. So we have 4 lines per piece, per every piece coming from the sequencing machine. So every read coming from the sequencing machine has 4 lines in the FASTQ file. So the first line is the ID, but the second line is the read itself. So this is what we are looking for, which is the second line in the FASTQ file. And the third line, just ignore it, can be only a plus sign or plus sign plus the ID. And the fourth line is the quality of every base in the read. So the sequencing machine will tell you how confident it was when it was sequencing every base. So these are an ASCII code. You can convert every character to ASCII code, and that will give you the percentage of the confidence. What we do, as I said, we need to build the index first, the index of the book. So we got from the reference genome pieces. We don't need the full reference genome. We need only pieces, exactly as we do in the yellow pages. And we store these in an index. The index is something that we can query or ask quickly, and it can reply back very quickly as well. So we need to store these pieces somewhere. Assume it's a lookup table or hash table or any kind of indexing. Now we got the FASTQ file, which is the person name, for example, the full name, which is the second line over here. Assume it's a full name of a person that we would like to get the phone number for. So we extract pieces from that again, because we will use these pieces to query the index that we just built. So we ask our index, do you have this blue piece somewhere? And to reply, yes, I have it in 2 times, over here at this location and over there at that location. Now I ask the same thing for the green and the red pieces. Do you have them in the index? To say yes, 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 I got the location of these pieces in the reference genome. Now that will reduce a lot the search space. Instead of having the brute force algorithm to search every single location, I'm just going directly to the location where these small pieces exist. And I need to make sense out of all these locations I get. For example, the blue, I got 2 locations, the green piece, I got 2 locations for it, and the same thing with the red. Now, how I make sense out of these pieces? I said the blue always followed by green, and the green always followed by a red in this read. So I need to find exactly same order in the reference genome. So whenever I have the blue, I check, do you have a green piece after this guy? No, apparently. So probably this piece here is wrong. I'm not interested in this piece, the blue one. I hope you can see my cursor. If not, I can use annotation. Okay, seems yes. All right, but when I go to the next blue piece, I find it's followed by a green and it's followed by red, which is exactly same order I have in the read. So probably this is potentially a very good location. And for that, I extract this subsequence over there and I start comparing it fully with the read sequence. That is, will give you the page number in the yellow pages book. And then I try to fetch the full name and then get the phone number. So the phone number or the final result of read mapping would probably be the comparison of the two, or the result of the comparison of these 2 pieces. What is the result? Is basically where they differ, where they match. For example, how this would be look like this. So maybe I need to delete all these, and then the T match this, and then the blue match that, and then I need to do some alteration over there, and so on. So every operation I do to make one of the pieces looks like the same, the other one, that is the result of read mapping. Okay, how we do that in practice? This is the reference genome. I chunk it into pieces. I store it in something that we call index. And based on our paper, we find that hashing is the most popular indexing technique for read mapping since 1988. So for the last 3 decades, it seems most of the paper or most of the tools are using hashing as indexing technique. So you use hash table, you store these small pieces there along with their location list. So every piece where it appears first. So that is 1. It means this seed appear first at location number 1. So which is the first character in the seed. It's not the location of the entire seed, but rather the location of the first character in each seed. Same thing with location number 9. It means the first character in that seed appears at location number 9, which also true with the second character in the seed appears at location 10 and so on. So you got the main idea, hopefully, from the hash table. So we store all locations for all of these seeds inside the hash table. This is a pre-processing step and it's done once. So whenever we have a reference genome, we build the hash table and that's it, done. Then we can store that hash table somewhere and use it whenever we want to do read mapping for human genome. Now, when I say seed, sometimes we refer to it as a k-mer. And K here is the length of that seed or subsequence. For example, if the red piece here is 10 long subsequence, then we call it 10-mer. So the K is the length of the seed, and we replace it with the length of that seed. This is some number about the index size, the indexing time, and so on, just to give you a hint about the workload that you are dealing with. Again, as I said, the index is built only once for each reference genome. And the seeds, the way we extract the seeds, there are a lot of theories, a lot of tools that extract the seeds in different way. They can be overlapping, non-overlapping, spaced, or adjacent, non-adjacent, and so on. So we overview all of these methods in our paper. So if you're interested in finding how it works, we recommend everyone to read it. So we compare here three state-of-the-art tools or well-known tools, and we find the index size really varies. So remember, the human genome, the file or the FASTA file size is 3.2 gigabyte, because we have 3.2 billion character, and each character is represented using single byte. That is 3.2 gigabyte. And when you build the index, you will see the size of the index is really huge, at least twice the size of the original reference genome. And the indexing time really varies. So to build the hash table, depending on how many seeds you collect from the reference genome, can be something between 3 minutes all the way to 1 hour. But don't worry about that, because it's still a pre-processing. It's not part of the read mapping process. So you build this once and then you load it into memory. The performance of a human genome indexing also varies for other tools. So I told you about building the hash table, but there are still a lot of methods for building the index. You can see Boris-Wheeler transformation, for example, over there, suffix tree, for example, another technique to build the index. So you can see the CPU time for hashing, again, really varies. Can be the fastest, but can also be the slowest. That depends on how you collect the seeds. If you collect non-overlapping seeds, it will give you a small number of seeds. But if you collect all overlapping seeds, it will give you a lot of them, way much more than the reference genome has. Boris-Wheeler transformation, it's well known for its fastness, because it's really fast to handle the reference genome, since it uses a compressed version of the reference genome. And the memory as well is really low. Memory footprint for the index is really low, since again it uses a compressed version of the reference genome. The suffix tree is not always the best. You can see in terms of memory and the CPU time, they are on average the worst compared to the other technique. Okay, so now assume we finished the index. Now we need to query that index. We need to reuse it. So we load it into the main memory, and now we got a bunch of reads. So we got read 1, read 2, read 3. And for each read, we collect some seeds. For example, here we collect 3 seeds, and we start to use them to query the hash table. When we query the green, for example, it gives you this location list. When you query the blue, it gives you that location list, and so on for each seed. Now we got the location lists for each seed from the read, and this is the reference genome. The one below is the reference genome. These pieces are the pieces from the read. So we need to make sense out of all these locations again and see which one respect the same order as they appear in each read. For example, here we have the yellow appears at earlier than the blue. So we check the yellow and the blue location, we sort them, and we find that 5 appears before 7 and 7 appears before 9. So it seems at 5, we got a good hit for this read. So we extract the subsequence starting from location 5 all the way to, for example, 100 locations if the read length is 100. And then we start to do another process to compare these two sequences. So we can query the hash table with the substrings from read to quickly find a possible mapping location. Now, the third step, once we figure out the good candidate location, the reference genome, we need to compare them using really thorough analysis. So we really need to find the differences at character level. So every character, how it's similar to the other character or the corresponding character in the read sequence. And what we use for that step, we call it sequence alignment or verification. And for that, we need dynamic programming algorithms, as you can see here. So we build dynamic programming table, and this is the read. This is the sequence from the reference genome. And we start to compare them together to get the exact number of differences and where they are and what type they are. So how we do that? We use, for example, one of the algorithm called edit distance algorithm, or Levenshtein distance, Hamming distance, or so on. There are a lot of algorithms can be used for finding these differences. They differ in their goal, in their methodology. There are also more complicated algorithms such as Smith-Waterman, Needleman-Wunsch, and so on. For edit distance, is defined as the minimum number of edits, for example, insertions, deletions, or substitution that are needed to make the read looks like exactly the reference genome or the reference subsequence that we extract. For example, assume we have organization, this word as a read, and the operation is a word extracted from the reference genome. Now we want to compare them and see how many differences we can get optimally. Because remember, the distance is about the minimum number of edits. So it should be the optimal number. Now, there are two ways to compare organization to operation. So you can, for example, you can use this legend to find these colors. For example, here we need to insert P and E to the other word to make it similar in this portion. And then we need to delete all these characters. And then the rest will look like exactly same as organization. Or you can do it this way. So you can alternate between exact match over here, over the A, and delete only the G, and then continue deleting the rest of the character until exactly match. Now, which one we pick, that is another question. And we use what we call alignment score to tell you which one is better. Because we don't know for this time, which one is better until you define which one you want to penalize more. Do you want more insertion or more deletion? Do you want more consecutive deletions like gaps, or do you want less of these gaps? So if you want less gaps, you can pick this one. If you want 2 gaps, you can pick this one, and so on. That's what we call alignment score. So you need to define this scoring scheme before you start doing this comparison. So I will send you an Excel sheet that is prepared by a very well-known scientists in this area, where you can play with this comparison using Smith-Waterman, for example. So you can just insert your quality score, and then it will tell you the differences between any given 2 sequences. You can play with that. So the same thing applies to any different example. So here you can consider organization and translation, for example, and you can see different ways to arrange these operations. Okay, so that will be the output of read mapping. And we store it in a file, we call it SAM file. So you can see here in the previous slide, it outputs in a file called SAM or BAM. So SAM is the decompressed version of BAM. BAM is the binary file that has all this information once you finish sequence alignment operation. Okay, and in our paper, we find that Smith-Waterman remains the most popular algorithm to do sequence alignment since 3 decades. And Hamming distance is the second most popular technique since 2008. So probably you are aware about Hamming distance, which is used for encoding signals and signal processing. So it was proposed by Richard Hamming, very famous scientist. And this Hamming distance is basically performing XOR operation between the 2 sequences. So just comparing every 2 corresponding characters together, and if they are different, then you calculate the number of differences in total for these 2 sequences. So it's very simple, but cannot find insertion and deletion. It will have a huge impact on the result if you try to find the differences between 2 sequences that have insertion or deletion. So the goal of this algorithm is really different from Smith-Waterman, for example. And this is another paper that my PhD advisor, Jan Khan, was working on in the past, where he built a very fast tool in that time, relying on hash table to do the indexing for these seeds. So I recommend everyone to go through it and read it. There is a new updated version of this paper called Fast Hash. So you can also check that. And both of them result in what we call Mr. Fast tool. So you can access Mr. Fast over there and check how it performs. Now, when we talk about the full read mapping, not just only the indexing step, we do the analysis for existing tools over the last 3 decades. And we see that the recent tool start reducing the processing time a lot, really considerably compared to the previous tools. And also on average, they maintain the same performance over different samples. So you can see these small dots, these are small different samples, different read file, different workloads. So that will tell you something about the need for accelerating read mapping. So in the past, we were not worried about the performance of read mapping because sequencing machines were slow anyway. So why I would do the analysis in 1 second if the sequencing machine is performing the sequencing in 1 day or in 1 week? And in the memory, a footprint really varies even until now. So you can see if you want to fastest performance, you need to maintain somehow high memory footprint, but also we have other tools that still perform good in terms of processing time plus the memory footprint, as you can see over there. So based on this result, we see that at some point, all the tools start to following a trend of having very fast read mapping operation. And for that, we did the analysis for all the tools following 2013. Why 2013? Because you can see after this point, we start to have what we call long read sequencing machine, or the 3rd generation of sequencing machine. At this point, we start to get very high throughput machines that reduce the cost of sequencing. So at this point, we start to get the 2nd generation, which are Illumina-like devices. Like these machines produce 100 long reads, but at this point, we start to get machines that produce million long reads, for example. So that will increase the throughput within the same sequencing run and thus reduce the sequencing cost per genome or per base. And you can see the CPU time, since we increased the throughput of the sequencing machine, all the tools now need to be very fast to cope with the sequencing workload. Okay. Now let's conclude the things we were explaining before. So read mapping is to map reads to a known reference genome with some differences allowed. I hope this definition is very simple, very clear to everyone, so you can keep remembering it even if after finishing or completing this course. So we got the sample, we got the reads, we map these reads to the reference genome, and we try to get the complete human genome after that. Now, another application is what we call Metagenomics Analysis. So imagine if you swap a sample from anywhere in the environment. In the train station, for example, you went to the door and you take a sample and you try to sequencing it. Now, you cannot imagine to have a single species in that sample. You will see a lot of bacteria, a lot of germs in your sample. And for that, you cannot use single reference genome to do the analysis. So you have a lot of them, a lot of DNA footprint in your sample. And when you do the sequencing, you'll get a real collection of many reads coming from different samples or different species. For that, again, you cannot use just single reference genome as we do in read mapping, but what we will do is to get a database of reference genome. So every species exists that we know, we need to have it in our database, and then we compare every read to all of these reference genomes. So we do this one by one. We may find some hits in this reference genome or in that reference genome, and so on. We keep doing that. Remember, even if all these read, the yellow pieces, map to this guy over there, we may get some of the reads, for example, the red piece over there, mapped to wrong reference genome. Why is that? Because we are still comparing short subsequences to the entire reference genome. And for that, the probability to find hit in all these reference genomes is very high, especially when we talk about a combination of only 4 characters, A, C, G, T. So when you enumerate the possible sequences that we can build out of ACGT, you will find a lot of hits in all these reference genome. So whenever we have shorter subsequence, the probability to find a match is really high when we talk about very long string. That's what we call metagenomic analysis, but I'm not going to explain this in this lecture. But if you are interested, please follow. Before following our papers, so this is another analogy to think about it. And read mapping, so it's like having homogeneous M&Ms, but when you are talking about metagenomics, you expect to find many different species, heterogeneous M&Ms, for example. So this is just an analogy to keep you remembering what is metagenomic analysis. Okay, so we have 3 different work with our collaboration from UCLA and University of Southern California. So the first one is MetaLine. If you're interested in this area, you can read this. Maybe you can brainstorm about it and find things to improve these tools. This is the second work, and this is the third work about metagenomic analysis. Okay. Yeah, again, as I said, the slides are already uploaded in the course webpage. It's publicly available for everyone, even those watching us on YouTube. So I'm going through the paper quickly. Now, let's talk about the challenges in read mapping. So first, we need to find many mappings of each read, because again, these are short subsequences, and we expect to find many hits within the reference genome. So they might map to location 5, plus 50, plus 5,000, plus 5 million, and so on. So we need to find all these locations and compare all of them to the read and find the optimal one, which we flag as primary alignment, and then the rest as secondary alignment. And they are useful for variation or genetic variation discovery and so on. The third challenge is that we need to still tolerate the genetic variation plus sequencing errors, as I explained before. And these 2 things are really important. Sequencing error, as long as we are still using machines, then we expect to have these sequencing errors. And the goal of this work is to have genetic variation, so we still need to use computationally expensive dynamic programming algorithm to correctly find the location of all these genetic variation. Now, the third challenge is that we need to map each read very fast. So performance is important, especially after 2013, especially in life-critical application or life-threatening application, where we really need to find the genetic variation that causes disease or life-threatening disease to critical ill infant, for example, admitted to hospital. So you really need to fight with time to find the cause for that disease or the illness, especially when the symptoms are not clear. So you cannot prescribe a medication without knowing what was wrong, so that you can have the correct medication or the correct course for each patient. And the fourth challenge is that you need to map reads to both forward and reverse strands. What does that mean? So as I said in the previous lecture, the DNA is really double helix structure. It's coiled. So this is one strand, this is the other strand. They are attached to each other, chemically binded to each other. Each base is connected with 2 or 3 hydrogen bonds. And when we do the sequencing, we need to split them away from each other. So we get this, what we call strand or forward strand, and this is backward strand. And you need to split them from each other and then start sequencing any one of them. So you don't know which one you are splitting or which one you are reading currently. So that's why we assume that we are doing read mapping to both strands, to the forward strand and the reverse strands. How is that? When we build the index, we build the index only for one of them. Again, we don't know which one is recorded in the FASTA file. So we assume that we have any of them, and we record it in the hash table, but also we can record some pieces from the other strand, given that we know that these are complement or reverse complement of this strand. For example, whenever we have a CG, then it should be GC in the other strand, because it's always reverse complement of one of the strands. So we can record both seeds from this strand and the imaginary one that we have in our mind, which we don't have this one normally in hand. So you can imagine it that you have it, and then record pieces from this strand and the other strand in your hash table. And when you find good hits in the other strand, then you assume the read is coming from the reverse strand, not from the forward strand. In this way, you can tolerate this problem, or what we call strand bias problem, that is already solved now. It's no longer a problem. It's already solved by just following the methodology I explained, where you have you apply indexing over the reverse and the forward strand together. Please let me know if this problem wasn't clear, or any of these challenges was not clear. Okay, I see a sign of okay from everyone. Okay, cool. Now, I told you about the analogy of this puzzle solving thing, which I like it very much to explain the things. But imagine that we have a reference genome for single individual that's representing a single human, right? Although we have some variation recorded in the FASTA file, but still the FASTA file that we are reading or building the index for is for single human, and we use it for the entire world. That is wrong, of course, and still it's already something proven in the research papers. Imagine instead of using one reference genome, we need to use reference genome for Europeans, reference genome for African, reference genome for Asian, and so on. And then you do the comparison with that. Then the problem will be more complex than what we compare with a single reference genome. That's what we call a field of pangenomics. Pangenomics is studying collection of individuals rather than single individual. It's different from metagenomics, because in metagenomics, you have species from really different organisms or different kingdoms. For example, bacteria, viruses, human, banana, onion, and so on. But in pangenomics, we consider a collection of a human only, or collection of bacteria, collection of viruses. And then we try to do the mapping to these different reference genomes. That will make the things really more complex. So this is one of the papers that try to do pangenomic study for African, and they find that African are really superior compared to other humans. They have 10% more DNA bases compared to the existing reference genome. So that proves that it is wrong to use the reference genome for all individuals in the world. For that, we really need to use customized reference genome for every single population in this earth. This is another paper showing that it's the time to change the reference genome. And for that, we start to see more tools building graph instead of linear representation of the reference genome. In the graph, you can show diversity. You can represent whenever reference genome differ between individual and individual, you can have different branches in the graph to represent such diversity. And so for that, the recent tools are more focusing towards graph processing. Okay, that was all for the step or the definition of read mapping, showing you computationally how you perform read mapping and so on. We show you a list of tools that perform read mapping, how they perform, and so on. I hope that was very much clear. Okay, I saw a sign of yes again. Thank you. Okay, so now when we do the computational analysis to see how read mapping performs compared to sequencing. So we have a sequencing that is done using sophisticated machine, but when we do the computational analysis at the CPU, how they perform, which one is faster these days? So we see that the sequencing is done really well, can process about 48 human genome in about 2 days. So every human genome can be read in 1 hour. That is really quick. But when we do the analysis at the CPU side, it's still bottlenecked by the CPUs, by the current CPUs, or what we call read mapping. So read mapping is very slow to be performed in current CPUs. For example, we need 32 CPU hours to just do read mapping for single human genome. And for these numbers, you can check this paper, or you can check many more white paper from ONT and Illumina machines. Okay, what makes read mapper slow? This is something interesting, especially if you are focusing on using GPU, FPGA, and other hardware acceleration platform. What makes read mapping a bottleneck in genome analysis pipeline? The first thing, we have a tsunami of sequencing data. So as I told you, day by day, year by year, the sequencing machine are getting developed. They are really advanced these days, such that they can generate a huge amount of data in a very cheap price. And that motivates us to start sequencing something that we never studied before. For example, in the ocean, we start to discover what is the reference genome for this kind of species or this kind of fish, just out of curiosity, because sequencing becomes cheaper and more available. We can do sequencing in space, for example. There are a lot of papers showing that. We would like to figure out what kind of species exist in space. So you can do sequencing over there and then analysis. That makes more reference genome available for other species. However, the read mapping is slow because, as I said, we are using specialized machine to do the sequencing, but we are still using general purpose machine for the analysis. We are using our general purpose computers to do the analysis, and that is normally slow because we don't have any sophisticated architecture for read mapping. We are still using the Neumann model that is from 1948, which separating the storage from the processing. So you have the storage over there, and then the main memory, then different level of caches all the way to the core to do the processing. For that, you still need to move the data between different components, and that is a bottleneck in the current analysis. So data analysis is performed far away from the data processing or from the data storage, and that dominates performance. So as you know, data movement between the sequencing machine to the storage, to the main memory, to the CPU, it's very expensive. So single memory request, just requesting some data from the main memory, consumes at least 160x more energy compared to performing an addition operation at the CPU side. So you can think about moving a huge amount of reads from the sequencing machine to the CPU to do the analysis. So that data movement dominates performance and is always a major system energy bottleneck, accounting for at least 40% of the total system energy. When we also go deeper, pick one of the state-of-the-art read mapper called Minimap2, and then do the analysis or the breakdown for the processing time. We find that about more than 60% of the time just spent in the last step, which is verification or sequence alignment. So it's a very expensive step to be performed because it uses a quadratic time algorithm or expensive algorithm to do the analysis. Now, if this step or sequence alignment is expensive, why we are using it? Can't we use just something simpler, something heuristic? So the thing, dynamic programming algorithm are really good. Why is that? Because think about this example. So you want to compare Netherlands and Switzerland. So you really need to pick Switzerland, compare it with S, with SW, with SWI, with SWIT, and so on. So you see that they are redundant computations. So you already compare with the S, but then you need to compare with the S again, and then with the W, and then with the SW again. For that, dynamic programming is the perfect solution to not repeat the same operation. How is that? When you compare N and S, you store the result in one of the cells over there. Now, when you compare the SW with the N, you already have the result for Sn, so you just need to compare W with the N and reuse this result to calculate this cell. That's why we need dynamic programming algorithms. So it's something, it's unavoidable. You cannot just go behind it and use something else. However, these kind of algorithm, they are limited in data dependencies. So you need really to go cell by cell so that you can reuse the previous cell. You cannot skip every cell and just jump everywhere to do parallelism. For that, you need to respect the way you do the processing. So you can go row-wise, row by row, or column-wise, or anti-diagonal, for example. There are other ways, other methods shown in the research paper other than these three methods. And that limits the parallelism. So you cannot accelerate it in FPGA using just calculating everything in parallel. Again, there is some order you need to respect. You need also to calculate the entire matrix, even if they are really highly dissimilar. If this sequence is dissimilar from that, you need to calculate everything until you calculate this cell over there, so that you know that there are 5 differences optimally, and then you decide if these are needed or something not interesting, so that you can ignore them. Now, this is a paper showing that mathematically you cannot do something better than sub-quadratic time, or you cannot really calculate it in strongly sub-quadratic time. So it's always need to be quadratic time algorithm to find the operation optimally. That's a very interesting paper from MIT folks. Okay, and the other challenge that makes read mapping slow, again, as I said, we need to find all location for every read in the reference genome. So we need to compare all these reads in every location in the reference genome where we show hits by some seeds, and then we do the thorough analysis for the comparison. To do better, you have the computing stack. So you can either target it from bottom or from top. From top, you need to do something with the data. So you can compress the data, for example, to solve the read mapping issue or the slowness of the read mapping issue. You can also improve the algorithms, or you can improve the software side, or you can go to the electrons or the logic and try to improve over there. You can build customized chip. It will be always better to build something that target the top and the bottom of the compute stack. So build a new algorithm plus a new hardware architecture. And this is also proven in our research papers from our research group. We have a lot of work in this area to improve genome analysis. Here, an example just showing you when you do matrix multiplication, if you use these programming languages, you will see the huge difference, a huge speed up. If you move from, for example, from Python to C, you can see about 50x of speed up just by using different programming language, same method, same algorithm, and so on. So that's why we encourage everyone in this course to start using C. It's okay if you don't know it, you can learn it because you have 3 months to do your project. If you move further, for example, to SIMD instruction, where you can do parallelism per instruction, so that is a huge speed up. You can see about 60,000x of speed up just because using software. All of these software techniques, we are not talking about hardware yet, although sometimes the SIMD units need to be implemented in hardware. But still, you use them as a software APIs. So for that, I hope I encourage you enough that we need intelligent algorithms and intelligent architecture that handle data very well. And for that, we need to introduce algorithm and hardware acceleration for genome analysis. So this is another paper from our research group showing all existing algorithm that was used, plus hardware acceleration that was used to improve the performance. Could be processing time or memory or accuracy of read mapping as overall step. And I'm showing here the 3 steps: indexing, pre-alignment filtering, and sequence alignment. And each of these, they have different type of algorithm that can improve the performance. So in the next lecture, I'm almost done with today's lecture. In the next lecture, we have 3 directions to talk about the 3 different steps of read mapping, starting from seed filtering or the indexing step, and then pre-alignment filtering, and then sequence alignment, And I'm going through this in detail in the next lecture. However, our contributions basically we were focusing on improving the CPU computation time by having specialized devices attached to the main memory, so they can replace the CPU, or they can do some specialized operation. And for that, we have many work. You can check them. I'm going through these in detail, each of them in the next lecture, by the way, and the next following a few lectures. Of course, we cannot finish all of them in one lecture. We also did something to do the processing near memory. So we don't want to move the data to specialized hardware, but rather enable the main memory to do the computation inside. So we reduce data movement, and for that we improve the energy plus the processing time, because it also takes time to move the data from here to over there. And for that, we have 3 different works. We also have some work about improving sequence alignment, which is dynamic programming algorithm. And we did that near memory or inside the memory device. That's just some motivation for you to follow up with the next lecture. And here are some pointers that you can check. This is our recent paper, other recent paper, another recent paper from our research group, and bunch of research talks on YouTube. You can follow up if you have some time. They are 30 minutes. This is 3 hours. And this is 1 hour and a half from Professor Nur Mutlu, and many more talks and research papers. That's all for today. I'm not sure if we have questions on YouTube or if you guys have any questions. Maybe I can stop sharing. Yeah, let me know if you have any question. I'm happy to discuss them right now. Can stay for a few minutes. All right, cool. I hope you are enjoying your projects. And I know some of you have lectures right now, another course. So thank you so much and take care. Talk to you next week. Bye-bye.
Bye, everyone. Have a good day.